Здравейте! Аз съм Силвия Коцева и в момента се намираме в лабораторията на гимназия Smart Science, където с нашите ученици ще ви демонстрираме един експериментален урок по биология. Okay, so I know that after this class you have a lunch, so let's talk about food. What do you know about a balanced diet? Is that the thing that is shown on the board?
Yes.
Yes? Can someone try to describe it? What is that? Valo?
A balanced diet is eating healthy food and sometimes unhealthy.
Sometimes.
Sometimes, not always.
Okay, any other suggestions? Mimi?
Having an equal diet with healthy food and unhealthy food, balancing them in calories.
Can you imagine a diet that doesn't have unhealthy food in it? Is it possible?
Yes.
Yes.
Okay, so actually, this is a balanced diet. Are there any groups of food that you can recognize on this picture? Vale?
We can see chocolate, lollipops, sweets.
Okay. Alexa?
We can see some fruits like bananas, grapes, apples, some vegetables.
Mishu, what do you see?
On the bottom right have some milk products.
Mm-hmm. As you can see, they're separated into different groups. Can you tell me how are they divided? Why? Why they are separated? What do you think?
They contain different things.
Different things, okay. Do you know how do we call these things that we can find in our food? Yep,
Nutrients?
nutrients. Okay, so do you know how many types of nutrients we have in our diet? Actually, a balanced diet is the right amount of each of that nutrients. So, we have three main types of nutrients. Do you know which are they? Eva?
Carbohydrates.
Carbohydrates. Do you know what is their function in our body?
We use them to make energy.
Okay. Another group? Mishu?
Fats?
Fats. What do we use them for?
To store the energy?
To store the energy, that's right. And the third group is?
Proteins.
Proteins. So, today we're going to focus on the third group, which is the group of the proteins. Before we start our experiment, we should take a look at the structure of these biological molecules. Their structure is pretty complex, but for the purposes of today's lesson, we need to take a look only at their primary structure. As you can see, they're large molecules made of smaller ones. These smaller ones are known as amino acids. Have you ever heard of them?
Yes.
They're pretty much the building blocks of all of the living things, including us. These amino acids are connected by special bonds, and when we eat foods that are rich in protein, we need to digest them, to digest the proteins, which is something like cutting these amino acids, so our bodies can use them to make its own proteins. And these bonds are known as peptide bonds. We will switch them and we will have a really colorful experiment. So let's start. You can see that you have 4 samples right in front of you, and with this experiment we will try to find which of them contain proteins and which are not. So this is a colorful reaction, which means that we will have changing what? Color. Okay, so, but before that we need to make something that is known as standard, which is the thing that we're going to compare the other results to. Can you tell me something that you're absolutely sure it has no protein? Mishu?
Water?
Water, okay. Actually, this is the first thing that we are going to use. Here, I have clear water. It's tap water, actually. It doesn't matter. We will put in here in the test tube. And after that, we're supposed to add our reagents. This is sodium hydroxide. Be careful with that. And then a little bit of this blue solution, which is copper 2 sulfate. And as you can see, the solution changes its color to what? Bright blue. Okay, this is our negative reaction. And now we are supposed to make a positive reaction. Can you give me an example of something that you know that has a lot of proteins in it? Elena?
Egg.
What?
Egg.
Egg, that's right. And egg white in particular. So we are going to use egg whites as our sample. Then I will add what? What is this?
Sodium.
Okay. And? Okay. Can you suggest the color that we are going to have here?
Purple.
Dark blue, purple? Why not green?
Because we can be purple on the board.
Okay, let's see. It takes a little time. We need to shake it. This is our positive reaction. What is the color?
Purple.
Purple. So now we have two things that we're going to compare our results to. So the positive reaction is purple because the copper is forming a complex with the nitrogen from the peptide bonds. Okay, so you have 4 samples and 4 test tubes, A, B, C, and D. We are going to test them to see if they have proteins or not. 2 of them will have and 2 of them not. After that, you're supposed to guess what do we have in these samples. So I'm putting the first one here, then I will add what? And? What is the color?
Blue.
Is it positive or negative reaction?
Negative.
Okay, let's compare them. As you can see, it's obviously a negative reaction. Okay, let's try substance B. Sometimes it might take a little bit longer for the color to change, but what do you think? Is it positive or negative?
Positive.
Okay, sample C. Positive or negative?
Negative.
Negative.
Negative, okay. And the last one? What do you think?
Positive.
Okay, so we have 2 positives and 2 negatives. Now let's start your experiment, and after that I will show you actually what are the substances that you have, and then you will guess based on the results. You ready?
Yes.
All of you?
Yes.
Okay, now it's time to check the results and I need a volunteer. Vala, come here. Let's see the colors that you have. The first one.
The first one is negative, and
Why it's negative? Because
it's blue.
it's blue. Okay, is it dark blue or bright blue? Lena? Take your samples. Take a look at them. What color is that?
Light.
Well, I can't say that it's dark, so I think it's light blue.
Yeah.
Well, not as light as the first one, but okay. So it's negative and it's light blue. Is it clear or cloudy? What do you think?
Clear.
Clear. Okay, so sample A is light clear blue. The next one, is it positive or negative?
Positive.
Positive. Why?
It's purple.
It's purple. Is it clear?
Yes.
Yes. Okay, so we have Alexa, clear purple color. Okay. The next one, Eva?
Well, it's negative.
It's negative. That's
Because
right.
it's light blue.
It's light blue. Okay, is it clear?
No, it's cloudy.
No, it's cloudy. Okay, so we have light blue, but this time cloudy solution. Okay, and the last one, positive or negative?
Positive.
And the color?
Dark purple.
It's purple. Okay, is it clear? Yes. Yes, it is. Okay, thank you. Okay, so now I will show you what are those four substances. We have gelatin, starch, protein powder, and sugar. Two of them are proteins and the others are carbohydrates. Which are the carbohydrates? Lena?
The sugar and the starch.
The sugar and the starch, so those are going to be our negative reactions, right? And they are A and
C.
Okay, do you know how can you distinguish the solutions between sugar and starch? What is going to be the difference? First, let's take a look at the results. A is light blue and clear, and C is what? Cloudy. Okay, so can you guess which is the sugar?
The sugar is in the first one because it's dissolved in water.
Okay, and C is?
C should be the starch.
Okay, write them on the back of your paper. And we have the two positive reactions
The gelatin
and the protein powder. Okay, here is something that you should know, and that's that the darker the color is, the purple one, that means that we have more proteins in that particular substance. So, which is the darkest one? Okay?
The darkest one is sample D.
Sample D, okay. Do you agree?
Yes.
Okay, and so this is going to be protein powder, and sample 2 is
Gelatin.
gelatin, that's right. So today's experiment was about proteins, one of the largest groups of nutrients, and next week we will continue with another one, carbohydrates. Това беше нашият урок с гимназистите от гимназия Smart Science, в който за пореден път си доказахме, че науката може да бъде лесна и интересна.